Well, hello everybody. In our group, we research loop mimics at protein-protein interfaces. And it's important to understand why. Let's say we have a protein receptor and it binds a protein ligand. If we'd like to displace that protein ligand and agonistically give the same response, we might make a loop mimic to bind in the very same site. Now that loop mimic, if it was just a peptide, it would have a horrible delta S, and the delta G of binding would be poor. So what we do is we constrain it into a loop, into a cyclic system, and therefore make a cycloorganopeptide comprising of a peptide part and an organic part. This is a real protein-protein interaction, one we're interested in. It has an interface loop, and it has an interface helix too. And we're also making helical mimics in my lab, but I won't talk about that today. The very first loop mimics that we made included a compound called D2. D2 is a mimic of a loop in a neurotrophin. Neurotrophins are a series of exogenous growth factors that bind to cell surface receptors or nerves. When they do, they cause autophosphorylation of the TRK receptors, and this triggers a cascade of signaling events which result in nerve cell propagation and survival. When the neurotrophins bind the TRK receptors, they dive in headfirst into the TRK, and they expose these three loops, loops 1, 2, and 3, to binding the TRK interface there. So these three loops are responsible for most of the affinity of binding, and therefore they're called hot loops. D3 is a hot loop mimic of nerve growth factor, one of the neurotrophins. D3, we patented it. A biotech company took it over. They got it to phase 3 clinical trials for the treatment of dry eye disease. What is dry eye disease? You probably felt it already. If you stare at your computer for too long, your eyes go dry. It's uncomfortable, but it's not very serious. But it's extremely common. It's one of the most common diseases in the United States, arguably the most common disease. And drugs on the market for dry eye disease, they treat the symptoms, eye drops, but they don't treat the cause. D3 agonizes TRK receptors in the cornea, produces more mucus, and therefore treats the cause, the lack of mucus in the eye. Now, we were impressed with how far D3 got, so we made another series of compounds recently, also cycloorganopeptides, and they included two, one called C1 and one called PAN, which show a lot of promise. We teamed up with an expert in dry eye disease, a lady called Cynthia at Baylor College of Medicine, Department of Ophthalmology. Cynthia devised a model for dry eye. On the left here, you see a normal cornea for a live mouse. It's stained and it looks healthy. In her model for desiccating stress, she blows softly air at the corneas of mice still alive, and the mice cornea gets dry. And when you stain it, you can see it's perforated, it's irritated, it's not healthy. Simultaneously, in that experiment, she does the same thing with another mouse, but then treats with eye drops containing D3 or containing C1 or PAN. And what you can see here are the images for PAN. Heals nicely. What happens with C1, which I haven't shown, heals nicely too, equally effective. What happens with D3, in this assay, not active at all. That indicates that PAN and C1 are more efficacious in dry eye disease as indicated in her model. It doesn't mean that D3 doesn't work. It just means it's not as good a compound as C1 and PAN. Dry eye, if it goes on for too long, chronic dry eye may turn into something much more serious, and that's neurotrophic keratitis. In neurotrophic keratitis, patients' corneas turn green and they start to go blind. It's a very serious condition. There is a cure for this though, And the cure is nerve growth factor. Nerve growth factor, humanized, really expensive drug, but it can be used to treat neurotrophic keratitis. So let's recap then. D3 is a loop mimic of nerve growth factor. And D3 works in dry eye disease. It's logical that D3 may work for neurotrophic keratitis, but as far as we know, it's not been tested. C1 and PAN are more effective than D3 for dry eye disease, and they've never been tested also for neurotrophic keratitis. There's a lot of potential there. There's another compound class that's making us really excited in the lab. It came about because we're interested in BODIPs, and we were studying this SNAR reaction of this dichloro-disulfonated BODIP. It works really selectively and smoothly for sulfur electrophiles, but not for nitrogen or oxygen ones. So we did something outrageous. We took a peptide with cysteines on either end and containing lysine and threonine or serine residues, and we let it react with this BODIP in solution at 25 degrees for about 10 minutes. And beautifully, the two sulfurs react selectively and form a ring. So we formed a ring now with a cycloorganopeptide composition, but it's special because the organic part is a fluorophore. These BODIPs are bright green, stained, very fluorescent compounds. We can use that to detect binding to cell surface receptors like TRK. You can't do that unless you have some kind of handle, perhaps radio labeling, but it's not very convenient to do that. But with our cyclofluoropeptides, we can measure KD quite conveniently. And it's much better than isolating that cell surface receptor because it tends to misfold when you do that. So even for your techniques like SPR and BLI that some of you might know are much more accurate if the cell surface receptor is misfolded or only partial structure, then the data that you get are not very good. It's much better to measure the native receptor in a live cell. So we can do that, and if the cyclofluoropeptides are imported inside the cell, we can track where they go as a function of time and which organelles they localize in. Another big advantage. So cyclofluoropeptides enable early stage assays for probe discovery and for therapeutics. We discovered a compound called 5C1. It was discovered by a visiting student from Thailand, Dor, who began at Texas A&M this year as a graduate student. And we call it Dor's Amazing 5C1 because of this assay performed by my collaborators, Cedric and Arthur, at the Health Science Center at Texas A&M. This assay features, first of all, a control mouse. Its spinal cord was damaged, it was given a recovery time, and the poor thing, it can't walk properly. It's dragging its hind leg. It's permanently damaged. Here is another mouse. It was given the same injury at the same time, but treated with 5C1 every day. It's running around like a healthy mouse. You can't tell the difference between this and one that wasn't injured. That is amazing. It's even more amazing because the compound was injected, IP, into the muscle, and it found its way across whatever barriers into the spinal cord and caused this repair. So imagine for a second that you were paralyzed in a car accident, or imagine you was a soldier caught by a roadside landmine and desperately injured. Drugs like this would be very, very precious, and there are none for humans. So 5C1 has a lot of promise. But there's other things we must do in research. We must be able to scale up the synthesis of 5C1 so we can provide lots for animal studies. And that's difficult to do if you have to prep HPLC. So I've got an idea for doing that. We'd like to be able to increase the blood residence time for 5C1. In other words, make its pharmacokinetics more favorable. I've got a very good idea for doing that. And we'd like to also be able to increase the affinity of 5C1 for the TRK receptors. And I've got an idea for doing that. All we're working on at the moment. Ultimately, we want to be able to replace the fluorophore with something that's not fluorescent. because drug approval agencies are simply not used to seeing fluorescent drugs. So to do that, we can put in other organic things, and we've tried a little bit of this too. But there's a problem, you see, because when you make that type of substitution, you change the shape of the loop, you change the characteristics, and it may not be active. Fortunately, we have a secret weapon. And it's not a secret anymore because we published it in Anger Biochemy in 2023. It's called backbone matching. Backbone matching is a computational virtual screening technique that enables us to select, in cases like this, an organic fragment that could be used to substitute for that fluorophore and retain the same conformation. So we're using backbone matching at the moment to suggest organic parts that we can put in these cycloorganopeptides to retain their activity and get rid of the fluorescence, even though it was so useful for the early stage assays. Thank you very much for staying with me to the end of this video. Thanks for listening. Bye-bye.